any questions still with for the other speakers of this section that the audience and the floor might want to raise. So if you have any questions for other speakers, please feel free to do so right now.
Hello. So I do have a few questions for Justin.
Please.
Yeah, from Academia Sinica, Ziqingmen. So Justin, the talk was really interesting. I have two questions for you. The first question is there any safety concern for the host if you deliver bacteria phage to the gut? And the second question is, what's the half-life of the phage in the host body? Is that going to be frequently delivered, or you just need one shot? Thanks.
Thank you. That's very good questions. So introducing bacteriophage to the gut, there's lots of different types of bacteriophage. Some are lytic that can destroy or kill the bacteria. These are the ones we use are not of that type. These live harmoniously with the bacteria, and they do slow down the growth marginally, but they reside within their non-lytic form of bacteriophage. So they do not incorporate into the host genome. They replicate through rolling cycle replication of the bacteriophage genome, and yeah, there's not a notable difference that we see in how it impacts the communities, though we do see that in culture that there's a little bit lower, slower growth rate because they have to produce the bacteriophage as well as the enzymes that we've incorporated in the bacterial genome, or sorry, in the phage genome. The half-life, we looked at the study of the E. coli Nissl probiotic, and we've looked at introducing the bacteriophage. The bacteriophage clear very quickly, and the probiotic Nissl carrier that we used, that would clear within a few days, unless we provide some type of an advantage for it to be persisting. And so we could do that by, we tuned several factors. We looked at applying the probiotic over an entire week, an entire month, or just a few days. And by doing that, we could increase the persistence of it in the gut. But really, this is intended to be something that would be taken more frequently. For instance, once every few days, because 3 to 10 days was the lifetime of these.
Thanks. That's very informative.
Great. So there is a question from the floor. Am I right? There's a question. What is the mg of the vitamin C used in the study and the duration before it's effective? So it's a question directed to Masahito.
The question is in our epidemiological study, and in the Nakajima cohort, it's a observational study. They took vitamin C from foods. They don't take. They did not take supplement, vitamin C supplements. They took vitamin C from foods. So it's an observational study. We did not administer vitamin C.
Sure, thank you. Is that has that answered your question?
Yes.
Okay. So thank you, Masahito. And another question from the floor is, what are the regulatory considerations of bacteriophage-based manipulation of the microbiome for aging? I think it's a question directed to Justin.
Oh, thank you. That's a very good question. So there's a few different factors. One important thing to consider in designing it is that you would you don't want to have a permanent change of your gut metabolic activity. You don't want it to be something that's irreversible. And so that's important actually, that these clear out every few days. And another factor is that usually when one is preparing bacteriophage, a lot of the processes involve the use of antibiotic selection when you're doing your production of your bacteriophage. And so we've used a technique that we don't utilize any antibiotics. So we're not introducing antibiotic resistance into the phage. And so we're not going to be introducing antibiotic resistance into the gut. So you don't have to worry about producing any superbugs, so to speak, that are resistant to our different antibiotics that we have. Other factors could involve whether or not the bacteriophage are causing dysbiosis, like are they destroying bacteria in the gut? And these again are natural bacteriophage that have received a natural enzyme from a different bacteria, the bacteria that doesn't persist in one individual's gut, but persists in another individual's gut. So it would be analogous to maybe kombucha or some type of drink where you drink bacterial components. Not everybody drinks kombucha, but it's a probiotic type of drink. And so you can imagine there's lots of different types of beneficial bacteria, but these are bacteriophage, which are just vehicles for delivering natural genes to the gut bacteria that already exist, rather than introducing new bacteria.
Interesting questions, and thank you for your answers. And I think there are still some time for us to introduce ourselves and for the audience to share with each other. So are there any? Shall we actually there are 34 people in this room, and since we know the speakers already, shall we ask anyone who would like to introduce yourself? Anyone in this group who would like to show up with your camera and introduce yourself very briefly? Just tell us your name and your affiliation and your interests, research interests. Anyone who would like to share a little bit of yourself?
Hi, Jacqueline. Am I clear?
Yes.
Okay.
That is, you are, how should we call you?
Yes, it's difficult to pronounce it. I'm Qinshen Li from National Taiwan University Hospital from Taiwan. I'm the awardee of Academica Sinica of Taiwan. I'm actually a neurologist who is very interested in Parkinson's disease. So I'm quite interested about Justin's work, because it seems that bacteriophage-based engineering could do a lot of things, not only affecting the gut, but also maybe the brain function through the gut-brain axis, maybe to mitigate or slow down the neurodegeneration process of Alzheimer's disease or Parkinson's disease. So I know that the question time is over, but maybe Justin, would you be interested to share, maybe you have some kind of collaboration with your bacteriophage treatment in disease models?
Yeah, I think that'd be really great. I haven't really looked into too much in terms of how it relates to the gut-brain axis, but I am really interested in that area, potentially expanding into how we can introduce other types of small molecule hormone analogs or even peptides that are produced by introducing these genetic systems to the gut bacteria. So yeah, that'd be a really great collaboration. I would be happy to chat more with you about that.
Thank you.
Thank you, everyone. And since we just have a few minutes, would our collaborator, Professor Lee, you would like to shortly share about our project as well, very briefly? Victor? Hello, Victor? Okay, so maybe we should introduce a little bit about, I should introduce a little bit about our project as well, because it is quite related to the discussion today. We are working on the biomarkers identification and drug repurposing for the treatment of AD, Alzheimer's disease, using AI-driven techniques. And our project is in collaborations with other international collaborators. So the interest, something that interests us is about the ethnicity. So with, like today, Justin just mentioned that dietary preference could actually result in different disease outcomes. So we wonder whether you have any idea about how the dietary preference would also affect our Alzheimer's disease outcome. If you have any idea, please, we would welcome your sharing. So anyone like to comment about this or not? So one thing that is interesting is Masahito work on the vitamin C, right? So our question is about how your work, how many subjects have you done that on the human subjects? The experimental study is applied on humans, and if so, then do you expect the outcome would be the same or similar from as compared to the mouse models?
Yeah, thank you very much for your question. We have not yet obtained the result of mouse study, and we will see our mouse model. Our mouse model can replicate result of human observational studies, but we need mouse model because we need to know the precise mechanism underlying the risk of ApoE and female interaction between them. And I think the study will lead to a discovery of new therapeutic target of AD.
Okay.
Yes, I hope. Thank you.
Thank you, everyone, for joining this section. And lastly, I want to, I forgot to acknowledge my student who is one of the awardees of our project, Mr. Han Yang, who has been with us, and he has brought in a lot of interesting ideas. And thank you, everyone, for joining today's section. And we hope to keep in touch with you in the near future. Thank you.